with the value of 2, indicating that all these turtles are herbivorous, and we don't have any evidence for carnivory. Therefore, we can be certain that the range we observed in the nitrogen isotope values here is not due to differences in trophic level, but rather we have differences at the base of the food web. And then I'd like to discuss one application of using the isotopic niche to assess population structure. And here, because we observed spatial variation among the foraging sites, we might be able to use the niche to assess the connectivity between breeding grounds and foraging grounds. And here I'm using the example of Nicaragua. So I've combined both northern and southern Nicaragua into one polygon. And in this case, 66% of the Tortuguera turtles overlap with the Nicaragua polygon, indicating a maximum contribution of Nicaragua to the nesting population. So there have been a number of other methods that have also tried to assess the Nicaragua contribution to the Tortuguera nesting population, because these data are really important for management strategies and population modeling. And using tag returns, this estimate has been estimated to be, or this contribution is estimated to be 82 to 86%. And the disadvantage of this classical method is that the tag return data come primarily through fisheries, and therefore there's a bias for the capture effort. Satellite telemetry is limited by the expense, and of the 15 green turtles that have been tracked from Tortuguera since 2000, 80% have returned to Nicaraguan foraging grounds. Genetic mixed stock analysis doesn't have the same drawbacks as the tags and satellite telemetry, and using many-to-many approach, the estimate for Nicaragua was put to be at 65%. And that's very similar to our estimate using the isotopic niche. And therefore, this approach may provide a complementary method to assess population connectivity. So in conclusion, I found that Tortuguera green turtles are herbivorous and feed over a wide geographic range. The primary producer values, velocity and tactitinum, differ in their stable isotope values across the Greater Caribbean. We also found that the wide isotopic range we observed in the nesting population is influenced by xenopoietic factors. And therefore, we caution that stable isotope data from a highly dispersed population can be difficult to interpret without additional information about the base of the food web or using another approach like compound-specific analysis. And then finally, the spatial differences we observed in the isotope values among foraging populations may improve our ability to assess population connectivity between nesting and foraging populations. And I'd like to thank the following sources of support and people who helped out with this project. I believe I have time for questions.